All right, so welcome back to our Chalk Talk seminar series presented by STEM Fellowship. It is my great pleasure to introduce today's speaker, which is Jenny Zhang from University of Calgary, Faculty of Kinesiology, and she's going to tell us all about muscle mass and physiological responses during exercise. Take it away, Jenny.
Thank you, Liam. So, hi everyone. I hope you guys are all doing well and staying safe. Thank you for listening to this presentation. This is my first time participating in the STEM program, and I'm really excited to share my research with all of you young scientists. So my name is Jenny, and I'm currently in my second year of my Master of Science at the University of Calgary. My research is in the Faculty of Kinesiology, which is the study of human kinetics. So kind of how we move, how we exercise. And so today I'll just talk to you a bit about the kind of research that our lab is interested in, what we do here, and I'll present a study that we are actually conducting currently. So firstly, one of the overarching themes of kinesiology is this concept of exercise performance. So how do we perform exercise? How can we be better? Why do we perform the way that we do? And to first understand this question, it's important to know that there are many factors that can influence performance. Here I'm going to go through only a few examples out of hundreds. So we can have environmental level factors such as altitude, which affects the concentration of oxygen in the air, and other things like temperature and climate. We can have characteristics that are specific to the exercise task that we're judging the performance on. So how intense or how long the task is, or the pattern that the muscles are contracting. So are we running? Are we cycling? Are we doing a bicep curl? And we have individual specific factors as well, such as age or fitness level. And all of these factors collectively can affect physiological and perceptual responses, which I will now discuss just a bit more in depth about what they mean. So in our lab, we have three primary physiological components that we're interested in investigating that can all influence exercise. So first, we can look at the cardiovascular system, so the performance of the heart, the blood circulation, oxygen delivery, and extraction. We can also look at the level of the muscle, such as different types of muscle fibers, or the mitochondria, which are organelles that can tell us how well we're utilizing oxygen and how much energy we are actually producing. And lastly, this is actually the main interest of the research in the lab that I'm involved in. We want to examine the nervous system. So I will expand on this one shortly, but for now we can think of it more specifically as a link between the brain and the spinal cord and the muscles. I also mentioned that there are perceptual or psychological factors that influence exercise. The importance of these factors are reflected in, for example, how athletes develop coping strategies to deal with their training, or how they get themselves mentally energized or pumped up before a game. So a few examples of perceptual factors that we can measure include effort, motivation, fatigue, or tiredness, engagement, breathlessness, and pain. So now let's talk about the main interest of our lab, which is fatigue and the nervous system. And we're interested in learning more about fatigue because it's an unavoidable component to daily life activities. So for any sort of task, we will experience some degree of fatigue. So what is it exactly? Well, a simple definition of fatigue is an exercise-induced reduction in muscle force output. So basically, when we exercise, fatigue occurs. Within our body, fatigue comes from the central nervous system, which is the brain and spinal cord, and the peripheral nervous system, which are the nerves at the rest of the body, specifically those nerves that connect to muscles. And how do we measure fatigue? Well, we can look at force. So for example, if we saw how hard somebody was able to kick their leg before exercise, so this black line here, and then they performed some sort of exercise, and then they kicked as hard as they could again afterwards, which is this green line, we would likely see that force be reduced. So when I say we want to study fatigue, we basically want to know how a bike ride, for example, affects the muscles and the brain system that controls the muscles. Now let's try to delineate the difference between when we experience fatigue at the central versus the peripheral nervous system level. We can take a look at this model showing that fatigue is kind of like a loop. So first, the brain up here sends down signals telling our muscles to exercise. If there's any sort of decrease in the neurons at the level of the brain and spinal cord, like for example, if the neurons are firing more slowly or less of them are firing, then we would call this central fatigue. So these signals eventually will reach the muscles, and our muscle is essentially like a machine that generates force, and it has a lot of components working together to generate that force. If there's any sort of changes to any of these components that would impair this force output, we would call this peripheral fatigue. And to close this loop, of course, so as the muscles exercise, they send information to other neurons, and that goes back up to the brain. And it tells the brain, okay, we're exercising, maybe we're getting a little bit tired. And eventually, as peripheral fatigue in the muscles build up, the brain starts to feel a little bit threatened, and it doesn't want to send more signals down to make the muscles even more tired. So to reiterate, brain sends down signals to muscles, muscles send signals back up to the brain. This is the general idea of fatigue. Hopefully it gives you guys a somewhat clear intro into this concept. So before I delve into the specific work that we're doing in our lab right now, I want to introduce this framework that we like to use that kind of summarizes the main idea of everything I just talked about. So in general, when we exercise, we observe certain responses in terms of heart function and changes in the neurons and perception. But in turn, these heart, neural, and perceptual responses can also influence exercise performance. And additionally, these responses can influence each other as well. And as I introduce our lab research, I want you to keep in mind that even though each of these concepts individually have a lot of components and many research studies behind them, all of them also integrate together to give us a complete picture of human kinetics. So earlier, I said exercise performance is task dependent. I brought up a bunch of examples at different levels. But what makes things a bit more complicated for us researchers is that fatigue is also task dependent, and fatigue in turn affects exercise performance. So those same examples that I brought up earlier at the environmental, task, and individual level can be applied to fatigue as well. So here I have just one example. Let's say we're comparing two running tasks. So Usain Bolt holds the 100-meter sprint record, and Eliud Kipchoge holds the marathon record. Both of these guys are obviously extremely trained, but even so, can we say that at the end of each of these tasks, these athletes felt the same level of fatigue? The answer is likely no. A sprint versus a marathon are different intensities. They're different durations. Different things happen in the muscle and the brain. So it's more than likely that different patterns of fatigue occurred. So this is why we generally say that fatigue is also task dependent. So one of the individual specific tasks that we can look at is the amount of muscle mass that someone has. For example, if we were to look at amputees, can an amputee perform a 20-kilometer cycling trial at the same level as someone who has all of their limbs? If not, why? What difference does the amount of muscle mass we have available to exercise make? So this is the focus of the project we are currently doing in our lab. How does different amount of active muscle mass affect exercise performance, physiology, fatigue, and perception? From a cardiovascular point of view, there's a lot of literature that has already investigated this question. And generally they agree that if you have a smaller muscle mass, you will actually perform better. And I'll explain why. So normally we would cycle with two legs, right? So let's say that there's a maximum limit of blood and oxygen that our heart is able to pump. So when we cycle with two legs, the heart needs to pump equal amounts of blood to each leg. Theoretically, when your heart cannot pump enough blood to the muscles, this is the main reason that we get tired and stop exercise. But if we're cycling with just one leg and the other leg is just resting and it's not doing any work, then that blood that we would normally be giving to the second leg can instead all be used for the leg that's exercising. So the single leg that's exercising has all of this extra blood and oxygen to do work. It's not limited by the abilities of the heart. So now, if that's the case, if you get tired when you're exercising with only one leg, then that means there must be some other reason that's not the heart, some other factors that make you tired and stop exercising. And in general, we would say that those factors are muscle function and the nervous system. So to look at this from a fatigue perspective, I'm going to bring up an analogy of a factory. So a factory works to produce items such as cars or clothes, and as it functions, it creates pollution. And if the pollution stays in this factory and it doesn't leave, it affects the ability of the factory to work properly and make more cars. This is similar to our muscles. So when our muscles exercise, they produce all sorts of molecules and products that we call metabolites. And eventually, just like the pollution in the factory, these metabolites will build up. So this information that there's a buildup happening in the muscles is sent to the brain. So if you have a certain amount of muscle mass, you'll send a certain amount of signals to the brain. And if you keep exercising, your muscles will keep sending more signals and your brain will start to feel threatened. It's going to say, hey, I don't think this is a good idea. This isn't a good feeling for us. Let's maybe slow down or stop this exercise before something really bad happens. But if you have more muscle mass, you will send more signals to the brain. So that point where the brain feels threatened, it's going to happen earlier. So the more muscle mass you have during exercise, the more feedback signals will occur. And due to these feedback signals, the earlier you'll stop exercising. So this is the current working hypothesis that researchers have developed in terms of the relationship between muscle mass and fatigue. So like I said, there aren't that many studies that have looked at muscle mass and fatigue. The ones that have only used simple exercise tasks, such as a knee extension exercise that you would normally use at the gym, where you're kind of just sitting in a chair and swinging your leg up and down. This kind of simple task, it might be tiring for the legs eventually, but they're only using one muscle group. So we wanted to use a more vigorous exercise, such as cycling, that activated multiple muscle groups. So that was one of the first novel points of our study. The second novel aspect is, like I showed you guys before, we wanted to integrate physiological and perceptual parameters together. So fatigue plus cardiovascular plus perceptual. There really aren't a lot of studies that have considered measuring perceptual responses with different muscle masses, but we know that they're an important component to any exercise task. So it is simply not enough to explain any sort of responses or results that you would get by looking at only one type of variable alone. So by using an integrative approach and expanding our measures to be more robust, we can get a lot more information and insight into our research question. So what exactly is our research question? Well, the purpose of our study was to assess the effect of active muscle mass on fatigue alongside cardiovascular and perceptual responses using an exhaustive cycling model. Okay, so let's discuss how we went about conducting the study. Firstly, we ended up having a sample of 12 healthy, recreationally active male volunteers who were between 18 and 40 years old. They had a pretty good range of fitness levels, so some of them were a bit more trained than others. And we had 3 sessions in our lab. So in the first session, familiarization, they just came in and we talked them through the study, kind of showed them what we were going to do. And in the second and third sessions, they did the same cycling task, which we call a ramp. So a ramp is kind of like if you imagine cycling up a hill. So basically, you start easy, but the intensity gradually gets higher and higher, and you keep going until you physically can't anymore. So that's what we refer to as the point of exhaustion. So they did this cycling task once with both legs, like normal cycling, and once with only one leg. So the leg that wasn't cycling was just resting there. This way we have 2 different sizes of muscle masses that are activated, but we are still preserving the type of exercise task. And at the same time, this is a very tiring task. So the participants end up really gasping for breath by the end of it. And in terms of our measurements, We measured central and peripheral fatigue before and after the exercise and cardiovascular and perceptual responses throughout. So first, let's talk about how we quantitatively measure fatigue. There are three main components we look at: maximum voluntary contraction, or MVC, voluntary activation, and resting twitch. So what we do is our participants sit in a chair and they have an ankle strap that measures their force that's produced by the leg muscles. So to get an MVC, we simply tell them to kick their leg as hard as they can and their force go up. This is kind of the max force that they are willing their muscles to contract. To measure voluntary activation, what we have to do is attach them to a wire, and we actually give them an electrical shock to the leg. This shock is kind of like an artificial kick. So it forces the leg muscles to just really quickly contract. So when people kick as hard as they can, we shock the muscle during that kick, and if the muscle produces a little extra force by the shock, which is just this little peak up top here, we call this the superimposed twitch. So this tells us that there's still some reserve capacity. So people were still able to kick harder, but for some reason, the brain was holding back or it was unable to. This is what we call voluntary activation, and it's a measure of the central nervous system. And lastly, after the MVC and the superimposed twitch, we tell them to stop contracting and to go back to rest. And at rest, we shock the muscle once again, and this tells us the current state of the muscle machinery. So if we do this before and after they exercise, we can see how much muscle function got impaired. And this is called the resting twitch, and it's a measure of the peripheral nervous system. So next we have our cardiovascular measures. So you can see from this image that when the participants are cycling, they have a face mask that they are breathing into and out of, and that mask is able to analyze factors like how much oxygen you're breathing in and how much carbon dioxide you're breathing out. They also wear a heart rate monitor, and we also prick the fingertip to take a little blood sample before and after exercise to measure changes in pH. And in terms of perceptual responses, we had 4 in this study. So first we have perceived effort, which is how much the participants feel like they're pushing or how hard they're working. Then we have perceived overall tiredness, then how much pain they feel in their legs, and lastly, breathlessness. So as they exercise more and more, they'll start to hyperventilate and breathe harder. So let's get into the results. What did we find? So we can start off with general performance. So the amount of time that was spent in the single leg and the double leg conditions actually did not differ. So the participants took the same amount of time in both conditions to reach the point of exhaustion. In terms of the cardiovascular responses, when participants were cycling with both legs, they had a higher ventilation and higher heart rate. So they were breathing harder and had more stress placed on their cardiovascular system compared to the single leg. Remember, this is kind of what we expected, right? So when people exercise with more muscle mass, there's more oxygen that's needed. So the availability of oxygen is a limiting factor. Now, getting into the fatigue results, let's start with the maximal voluntary contraction, MVC. So the blue bars was a double leg cycling session, and the orange bars was single leg cycling. For each condition, we have pre, so that's the lighter bars, that's before exercise, and post are the darker bars, so after exercise. And on the y-axis is the force. And then on top of each of the bars in each condition, I have the percent decline from pre to post within each condition. So after exercise in both conditions, participants were not able to kick as hard, but this percent decline was more severe after the single leg session than the double leg session. Voluntary activation, which is that superimposed twitch, if you remember, it also declined after exercise in both conditions. So after both double and single leg cycling, there was this reserve that the brain was holding back, but between conditions, there was no difference. And the size of the single twitch also declined after exercise. So after exercising, there were some impairments in the muscle machinery, but there was more decline in the single twitch after single leg cycling. So more impairments after single leg cycling than double leg cycling. So MBC was lower after single leg. Changes in the central nervous system was not different between conditions, and there were more changes in the peripheral nervous system after single leg. Okay, finally we have the perceptual results. So on the x-axis is the duration of the exercise task, so from beginning to end. For perceived effort, it increased as exercise went on and was actually similar in both conditions, but just slightly higher in single leg than double leg. Pain in the legs also increased throughout exercise, but it was again higher in single leg than double leg. Overall tiredness increased a bit throughout exercise, but it was not different between conditions. And breathlessness, as we expected, increased throughout exercise and was higher in double leg than single leg. We can compare this one to the cardiovascular response, where in the double leg session, there was a higher ventilation. So people were breathing faster and harder. So it makes sense that their perceived breathlessness would also be higher in double leg. So what can we take away from these results? I'm going to revisit this slide I showed earlier. So in double leg exercise, we saw that breathing was higher. So there was more stress placed on the cardiovascular system. The heart was not able to pump enough blood to keep supporting both legs. So that's the main reason why people stopped. But in single leg exercise, based on our expectations, heart function and oxygen availability was not a limitation. Instead, we saw that leg force and the abilities of the peripheral nervous system were lower, and leg pain was higher. So instead of the heart being the limiting factor in single leg exercise, it was actually the changes in the brain and the muscle that influenced how well people performed. So in general, we can take away that performance and fatigue are indeed muscle mass dependent. So what is next for our lab? Well, the study I presented to you today is actually part of a larger study that we are currently conducting. So instead of having our participants cycle at an increasing intensity, they're instead cycling at the same intensity at 4 different levels, and we're going to see the exact same measures and what happens. We can also extend our research question on how muscle mass affects performance to other populations, including clinical, such as amputees or athletes. And finally, we can explore options with how training can affect muscle mass and the impacts that might have. So yeah, to wrap things up, I have some people I want to thank. Acknowledging my supervisor, Dr. Jalal Abudarda. That's a picture of me and him in the lab. He's on the bike that we test our participants on. Obviously, this is before COVID. Also, thank you to my fellow contributors for the project and co-authors for our paper, as well as a big thank you to our participants for working so hard. Also, thank you to NSERC for funding and supporting our lab. And if anybody has any questions beyond today, please feel free to send me an email. Thank you for listening.
Wow, thanks. Yeah, really interesting presentation and easy to follow. So if it's okay, I'm going to get started with some questions. I'll start by asking you a couple of kind of like research, well, specific to your research questions, and then I'll maybe transition into some career questions, if that works.
Yeah.
So something that I noticed in the presentation that I thought might be interesting and maybe not immediately obvious to our audience was that you're measuring blood pH. So could you tell us, it might not be immediately obvious why that's relevant. And I just thought it'd be interesting. Maybe you could tell us a little bit about how pH is an indicator, might be an indicator of, physiological stress in this way.
Yeah, totally. So pH, I kind of call it as pH. It's more like lactate, but when you exercise, kind of depending on what intensity you're exercising, but what happens is you start to get, like I said, metabolites that build up, and one of those metabolites is hydrogen ions. And you also get a molecule called lactate that builds up, and accumulatively, they kind of just make the muscle a bit more acidic. And that's kind of just showing like there's processes that's happening in the muscle, and it's just kind of like a measure of fatigue. So if you measure pH from your blood, you can kind of get a measure of what state the muscle is in, how tired it's at, and eventually you'll probably reach a point where you can tell, like, okay, this person probably can't really go much longer, much further. Yeah, kind of like that. Does that answer your question?
Yeah, I think so. So, like, based on that, and this is more of a me question, but do you think that that data could sort of support your model that you've established, which is, when we're doing the single leg, it's not really about the physiology of the muscle, it's more about the brain's saying stop. Do you think that there's, like, does the data sort of, like, could you support that conclusion with the pH data?
um, it could be, theoretically you could. Based off of our findings, I think pH was pretty similar between sessions, just because in single leg, for example, you're building up your muscle, that it's not a lot of muscle, so it's working really hard. It's accumulating a lot of acidity, and then in double leg cycling, that maybe there's not as much stress in one muscle, but in terms of ventilation, you're breathing really hard. So it's like building up a lot of carbon dioxide, so it's still making it acidic.
Okay, right. So there's a little, maybe multiple factors there. Cool.
Yeah, exactly.
So this is, I think, at least of the ones that I've moderated, I think this is the first uh, Chalk Talk where the research has involved human participants. So this is, I think, a good opportunity for us to sort of get a little bit of insight into how that works. So could you talk a bit about, like, recruitment of, well, like, how human research works, what the extra requirements and considerations that you have to take when you're making that kind of research are, and anything else that might set this type of research apart from, like, cell work or something chemical.
Yeah, totally. Um, I personally really like human research. Cell work to me is, it's just cells, right? You can't really talk to them. You're kind of by yourself in the lab. Animal research I know is also popular, but I mean, personally, I cannot touch a mouse. Human research is interesting because it's a bit more relevant to us, but it's also more difficult. So especially in kinesiology, it's hard because we're telling people you need to exercise until exhaustion, until you're like really, really tired. So recruitment is really difficult for that sort of thing. In terms of ethics, I feel like it's also a bit more difficult because we are testing humans, right? Like, we're doing things to them. So you have to be a little bit more cautious about what exactly you're putting them through and what you're asking them. Another con would be that for human research, people usually have to give out some sort of incentive or like a gift to them, especially if you're asking them to do something really unpleasant. You might have to be like, okay, here's $20 if you do this for me. Whereas for cells and animals, you don't really need to do that for them. In terms of like results and data collection, human research I think is much more interesting. You get to like see things that are just more like relevant, like I said, and you get to talk to your participants. But the data is a lot more variable because it really depends on the kind of person, the kind of people you get. There's so many different factors that go through the human body, even if you're just investigating one little part of it. In cells and animals, it's a lot better controlled. Cells, you can just get any type of cell you want. It's more predictable, I would say. And with animals, it's not really as difficult. You can, have as many animals as you want, and they're pretty much all the same. So there's a lot of more challenging aspects to human research, but I would say it's just it's more fun and it's more interesting, especially if you're working with, if you ever do anything with clinical work or with patients, you feel like you're actually making like a cool impact. And yeah.
Cool. Yeah, of course. Okay, I'm going to ask a couple of more career-oriented questions now. So we just, right before this, heard from a research associate who already has a PhD. you're sort of at the other end. I think you said that you're in the second year of your master.
Yeah.
So could you just tell us a little bit about like what your career path, and to whatever extent that you're comfortable, what your career path has sort of looked like up to this point, what the plans are, and sort of like why you are, in the position that you're in at this time?
Yeah, so I did my undergrad at the University of Calgary as well, but I did it in biomedical sciences, which was uh, like very research focused. So I took a lot of classes that made us, read and write papers. I did summer research work in some like random labs. And I thought it was like the research aspect was like kind of interesting. I had a lot of labs where I did sort of like wet lab work with like cells and things like that. But I was like, I don't know if this is like really for me. And then in my fourth year of my undergrad, we do like an honors thesis, which is just where you have like, you go to a lab and then you do your own research project. You write like a big paper at the end and then you present it. So I did it in kinesiology with Dr. Martin McInnes, who is on the acknowledgement slide here. And it was kind of using this device where we measured, like we just kind of looked at the muscle, similar to this sort of stuff, but it was really, really interesting. And I was interested in kinesiology research because I started getting more into exercise and I was like, wow, like this is cool. I wonder what happens in my body. And then looking at athletes as well. So then I wanted to do a master's afterwards because I actually really enjoyed the research process. I really liked data collection. I liked interacting and testing the participants. I liked, looking at results and actually like forming your work into something cool. So I was looking at master's options, and my PI, Jalal, said that he had an opening for me. And then I was like, interesting, but his work I had never had any sort of experience in. I didn't even know what fatigue was. I didn't really have any neuro background at all other than like some class stuff. So I was a bit hesitant, but I was also really excited because his research seemed really, really cool. So far it's been really fun. I'm actually the only grad student in the lab because he just started as a professor like last year. So I'm the only student. It's, quite a bit of work because it is a new lab, but I like having this experience. I think, honestly, I'm like a lot happier as a grad student than an undergrad student because I get to do something that I actually care about, and also because I have time to do something. Like as an undergrad, I'm kind of just jumping from one test to the next assignment, and it's like very stressful and not great. But as a grad student, you have time to actually do what you want to do. And then after my master's, hopefully I get a PhD placement somewhere. I'm applying for funding now, so we'll see. But I really like the field that I'm in right now. So I would like to continue in it. There's lots of cool new projects that we're thinking about doing in our lab. So it would be great if I could stay here, but yeah, definitely would like to stay in academia.
Cool, thanks. Yeah, that nicely insightful answer. So on that note, about sort of the differences between grad school and what you were doing before, could you just give us an idea of what an average day at school or work looks like for you, and sort of like give us an idea of what to expect if someone might want to do something similar?
Do you mean in grad school?
More or less, like for you, if you when you go to work tomorrow, like what is your day going to look like?
Okay, tomorrow is an interesting day to me because tomorrow is the first day I'm testing participants again. So I'm actually really excited. I have a session at 8 o'clock in the morning in the lab. So for this study that I said, this is the next steps in our study, my participant tomorrow morning is doing a moderate session. So I'm going to come into the lab probably around 6:30 to set things up and just to make sure I haven't forgotten how to do everything. And then also obviously to clean everything. So my participant's going to come in and then I'm going to test him. Tomorrow it's going to be really long. I'm probably going to be in the lab for I think you'll probably cycle for 3 or 4 hours. So I'll probably be there for like 4 or 5 hours. And then sometimes, like before quarantine, when I was, in the lab more regularly, I would be doing back-to-back sessions. So I'd be in the lab many hours a day. But then after I'm in the lab, I'll probably come back up to my office and read some papers. I have a funding application I need to work on, some writing work that I need to do. And then, I don't know, you might also have like a class or something usually. So I don't have classes right now, but many grad students do have classes. And also this semester I'm TAing a class, so I probably would have to do something for that as well. And then afterwards I just get to chill.
Very nice. So you mentioned that, this group that you're working in is fairly new because your supervisor is only been recently appointed. And
Yeah.
that sort of makes me want to ask about sort of like non-research responsibilities and tasks that sometimes come with being a member of a research group. So could you speak to what other types of things other than, reading papers, writing papers, doing research, grad students and lab members tend to have?
Yeah, so I think like a lot of grad students have to kind of maintain the lab because supervisors are really busy with their classes and like with their own activities and things like that. So grad students basically maintain the lab space. So you have to, make sure everything is there if you need to order anything. Grad students will usually also have undergrads that they need to mentor. So last year I had one. This year I think I have two. So you have to like make sure their projects are going good. If they have questions, help them. If they're like, can you edit my paper? You have to do it. Other activities, I guess. I think, honestly, I feel like most of it is pretty much research-based. I mentioned TAing. I think most supervisors will get their grad students to apply to TA. So a lot of grad students do have that commitment. We also, our lab will organize like a journal club where we kind of meet once a week. We talk about papers. So as a grad student, it's kind of like my duty to organize that. And yeah, I feel like that's pretty much it.
Cool. Um, okay, we're running out of time a little bit, but I'll ask a few more. So you did mention here on your acknowledgement slide about the paper that you're working on with these collaborators. So for anybody that may not, be totally familiar with the academic process, could you just talk a little bit about what the difference is between, publishing your work versus writing your work in a thesis type of situation and sort of the importance of publishing for your career?
Yeah. So in a thesis, I think like as an undergrad, you take it like literally so seriously. You're like, oh my God, this is 40% of my mark. It's such a big deal. But a thesis is just kind of like, especially at the undergrad level, it's more just something that you write for your own knowledge and experience. But you do the whole thing, but you kind of just keep it between you and your supervisor and like some other people who will read it. For a paper, it's a lot more high quality. So if you're kind of like the lead of the study and you have to prepare the whole paper, you have to be a bit more in depth and precise. A thesis is also way, way longer than a paper. So in a paper, everything you say has to be like very purposeful and really high quality. You would also have to get it kind of approved between every co-author or contributor, and then you would send it off to whatever journal you want to publish in, and then you wait for people to accept it or reject it or give you comments, and it's a really, really long process. Whereas the thesis is kind of just like a long, really long paper that you would write for a class. And the only feedback you would really get is probably from your supervisor, and it's not really read by other scientists. But in terms of the importance of publications, it's like really, really important, especially if you want to stay in research or academia, because it just kind of shows like how much work you've done, what your productivity is, what your interests are. And it's just a good way to show people what you've done and what you're good at, basically.
Cool, thanks. And last question. So if for any audience or viewers who, are really intrigued by this sort of field of research, and especially with physiology and kinesiology, which we haven't had a lot of in this seminar series, could you give them, if they're really intrigued and interested, but maybe, we're talking to maybe like late high school, early undergrad stage, do you have any advice or sort of information that you want to share with them if they want to sort of get involved in a similar type of thing that you're doing?
Yeah, I would say a really good thing to do is summer research. That's how I became interested in the research world. So a lot of undergrads will reach out to a supervisor in some sort of research that they're interested in. And then for the 4 months of summer, so May, June, July, August, they'll have some sort of involvement in the lab. And even if, you can't be a summer student, sometimes the supervisor will just let you be a volunteer, and they'll let you kind of help assist in the studies, and then you get to see what they're doing and you get some responsibilities. So I think like a good thing to do, I guess summer is over now, but you could also just ask to like volunteer in the lab during the school year as well.
Cool, thanks. So with that, I think I will let you go. Before you go, I just want to say thanks so much for lending us your time and your knowledge. This is a great presentation, and I think that it will complement nicely some of the, as we were talking about earlier, some of the more microscopic cell and animal stuff that we've been seeing over the past couple of weeks. So I just want to let everyone know that the next, that we're done with our talks for today, and we'll be picking up again on the 13th, which I believe is this Sunday, at 12 p.m. Eastern with Lale Behat from the, she has many supervisors, so I won't read them all out, but she will be talking about understanding the world better through computational modeling. So look forward to seeing you there. And lastly, I'd just like to say one last thanks to Jenny. I really enjoyed this presentation, and I think everyone else did too. So everybody have a great day, and thanks again.
Thank you.
My pleasure. Bye now.
Bye.